Describe and explain the steps involved in PCR. 5 marks. The polymerase chain reaction, or PCR, is a cyclic process used to produce many copies of a specific DNA fragment. It begins with denaturation, during which the reaction mixture is heated to 90 to 98 degrees. At this high temperature, the hydrogen bonds holding the two DNA strands together break, causing the DNA molecule to separate into two single strands. This exposes the nucleotide bases so they can be copied. Next, the mixture is cooled to approximately 50 to 65 degrees for the annealing step. At this temperature, short DNA primers bind to their complementary sequences on a single DNA strand. These primers are essential because they provide a starting point and binding site for the DNA polymerase enzyme. The temperature is then raised again to about 65 to 75 degrees for the extension, which we call it elongation phase. This is the optimal working temperature for Taq polymerase, a heat-stable DNA polymerase. Taq polymerase then adds free nucleotides, which we call it dNTPs, to the primers and synthesizes complementary DNA strands, producing two double-stranded molecules from original one. These 3 steps, denaturation, annealing, and extension, are repeated many times, resulting in an exponential increase in the number of the DNA copies. Section B, Part 1. Describe how the TTAV gene could have been synthesized and transferred into the eggs of A. stephensi. 4 marks. To produce the TTAV gene, scientists first had to obtain the DNA sequences from E. coli and the herpes simplex virus. This could be done by extracting or cutting the DNA using restriction enzymes, or alternatively by making DNA from mRNA using reverse transcriptase, or by assembling the sequence from nucleotides. The two DNA sequences, bacterial and viral components, were then joined together using DNA ligase, forming the complete recombinant TTAV gene. A promoter sequence was added to ensure the gene could be transcribed inside the mosquito. The recombinant TTAV DNA was placed into a vector, such as a virus, liposome, or other delivery system. The mosquito egg was then infected with the viral vector, or the DNA could be physically delivered by microinjection or a gene gun. The viral vector then inserted the TTAV gene into the genome of the A. stephensi egg, allowing it to be inherited by the offspring. Section B, Part 2. Suggest how the TTAV protein prevents the expression of other genes in A. stephensi. 1 mark. The TTAV protein likely acts as a repressor, preventing transcription factors from binding to promoters or blocking RNA polymerase from attaching to DNA. This stops transcription and therefore prevents the expression of essential developmental genes. Section B, Part 3. Suggest why A. stephensi larvae were exposed to tetracycline in the laboratory. 1 mark. Exposure to tetracycline allows the genetically modified larvae to survive to adulthood. Tetracycline inhibits the TTAV protein, preventing it from blocking gene expression. This ensures that the modified mosquitoes can develop normally so they can be bred in the laboratory.